our today's guest, who is Professor Shuzo Sakata. He heads a research group within the Strathclyde Institute of Pharmacy and Biomedical Sciences in Glasgow, United Kingdom. His research, his team explores very interesting subjects because they study how brain states are organized at the neuronal circuit level, how they are regulated, and how they influence perception, cognition, and disease-related processes. So the central focus of Professor Sakata's work is to understand whether manipulating brain state can help uncover mechanisms of neurological disorders and potentially modify their course. And what is very fascinating for me is that Professor Sakata and his team addresses these questions by integrating very sophisticated methods of studies such as in vivo electrophysiology, fiber photometry, which we will be talking about today, optogenetics, and very complicated computational analysis. And this offers a system-level view of dynamic brain function. And this approach has significantly helped shape our understanding of how brain states relate to behavior and dysfunction. I would like to say a little bit more about the Professor Sakata background, because he earned his PhD at Kyoto University in Japan and continued his training as a postdoctoral fellow at Rutgers University in the United States. And now he holds a professorship at the University of Strathclyde. So among his recognitions, because he also got numerous awards, is the prestigious Sir Robin McClellan Award for Scientific Excellence. And what I would also like to mention here, he's also strongly committed to mentoring early career scientists, particularly those from interdisciplinary backgrounds. And I think it's a very important contribution that helps to shape the next generation of neuroscience researchers. So we are really delighted to have Professor Sakata here with us today. I'm very happy and very excited about the seminar, which I'm particularly interested in because we are also beginning our studies with fiber photometry. So without further ado, I'd like to give the floor to Professor Sakata and enjoy.
Well, thank you very much, Caroline and Claudia. So it's amazing. So just share screen then. Okay. Is that okay?
Yes, we can. You can see it, and we can hear you very well.
Okay. So thank you for this amazing invitation, and thank you for coming. So I know that most of audience are not familiar with fiber photometry, so that's why I'm here. So I talk about so what's the fiber photometry and how we are using in our lab. Okay. Yeah. Okay. So here's the outline. So there are 3 sections. So first, I will introduce fiber photometry, give us some basic information, and then I will give a little bit technical information. So if you are interested in fiber photometry and if you are interested in the introducing fiber photometry in your experiment, so the second section is about it. And then at the end, I briefly tell you about how we are using fiber photometry in our multiple projects. Right. Okay, so I think everybody is familiar with fluorescent microscopy. So how it works is as follows. So let's say if you want to see some brain sections, you want to see some neuronal morphology by expressing GFP or labeling some proteins. So first of all, you want to shine the light. So if you want to see green light, so typically we use a blue light. Then you will capture the emission light. So in this case, it could be green light. Okay. And if you do immunostaining, so there's a target proteins, and then you can use antibodies, and then you have some tag fluorophore. So if you shine blue light, you can typically see green light. Okay, so this is the essence fluorescence. So the fiber photometry is essentially the same. Instead of the objectives, so we will use a very simple optic fiber like this. So we will shine blue light, and then we will collect the emission fluorescent light. Rather than seeing this, so obviously you can see only so this is the tip of the optic fiber. So basically no spatial resolution. So it sounds boring, but so this technology can be applied in the living animal. So you can get much more richer and dynamic signals. So I made some movie. So let's assume that these are sort of virtual neurons, which expressing calcium indicators. So there are 100 neurons. And so this is a sort of tip of the optic fiber. And here we monitoring signal over the time. So let's play this. So a bunch of neuron firing, you get some dim light, but occasionally you get some strong signal like this. And then, yeah, so you got some very strong signal like this. So this is, so typically what photometry can work. Okay, so you got some rough idea. So just play this. So for example, so during this period, very few neurons active, so the signal is relatively dimmer, and you got some intermediate signals, and then much stronger signal like this. Okay? So this is how photometry works in the brain. So to do the photometry, you need basically two major components. So first of all, you need to express some biosensors in neurons or other cells actually, and you need some optical setup. So in terms of biosensors, so many people using so-called G-CAMP, which is a genetically encoded calcium indicators. So how this works is as follows. So there is a so-called circularly permuted GFP basically. So it's not functional, but so there are two extra domains. So basically this is the engineered protein. So these two domains came from calmodulin, which is the calcium binding proteins. So if calcium bind to these calcium binding domains, so these two modules will combine, and eventually this circularly permuted EGFP become functional. Thereby you can report, see when calcium signals happening. So this is how G-CAMP works. So there are a bunch of biosensors, as I tell you later. And another component is the optics. So by expressing, let's say, G-CAMP in some neurons and implanting optic fiber, you can deliver light by using laser light source. It can be laser or LED, and then you can excite these Frohofer, in this case G-CAMP. Then you can collect the light, the emission light, and then you need to detect the light. And then, of course, you need to control these devices with the data acquisition system. So this is sort of an entire photometry setup. So it's actually very simple. And so here I tell you the brief history of fiber photometry. So it started actually 2 decades ago. So group of scientists did the very first fiber photometry experiment in vivo. So what they did was, at that time, they didn't use a G-CAMP because G-CAMP was very weak. It cannot be applied in vivo. So instead, they used organic calcium dye. So basically, they label the neurons using calcium indicators. And then they implanted optic fiber. And then in this case, they use laser to activate neurons. Then they monitor population neural activities. Then 2010, a group of scientists used the G-CAMP at this time. This is the very first study when where the scientists apply GCAP for photometry. So they express GCAP in the layer 2-3 neurons. So in this case, actually, rather than implanting optic fiber, so they actually put the optic fiber over the skull by thinning the skull. So they put the optic fiber. So basically, non-invasive monitoring. So actually, they managed to monitor fluorescent signals. Then, so people keep developing this technology. So in this paper, they implanted up to 7 optic fibers across multiple brain regions, and they monitor calcium signals across multiple brain regions simultaneously. So not just that, so this is another innovation. So they used the multi-fiber array, much thinner, and they implanted the tens of optic fiber across multiple brain regions. So another development is this, which actually my group also using, so-called tapered fiber. So using laser and then changing the angle, how you penetrate the laser to the optic fiber, you can shine the light on the upper part of the optic tapered fiber or tip of the optic fiber. So thereby you can monitor fluorescent signals across multiple brain regions. So there are multiple innovations. And obviously, what we shouldn't forget is the development of genetically encoded sensors. So I mentioned about GCAP, but also scientists keep developing a wide range of biosensors, which I will explain later. But now we can monitor not just neurons, but also we can monitor neurotransmitter activities or neuropeptides as well. So basically, the technology, optics, and also these biosensors are sort of co-evolved over the past 2 decades or so. Okay, so that's our history. And if you think about in vivo measurement, so there are multiple options like electrophysiology or 2-photon or multi-photon microscopy. Or if you work in the zero field, some people using light sheet microscopy, or by implanting the so-called green lens, so the tiny objectives. So you can also monitor neuronal signals. So there are multiple methods. So compared to them, so the advantage or sort of strengths of fiber photometry is as follows. So it's easy to implement. And also you can combine with optogenetics or chemogenetics. And there are a wide range of applications, so versatile. And because you can target certain cell types, so you can express GCAP in some cell types, it can be cell type specificity. And because we use reasonably thinner optic fiber, you can penetrate the optic fiber deeply and less invasive compared to, let's say, this microendoscopy. This is sort of a weakness of fiber photometry. So as you can imagine, so we can typically use one single optic fiber, so it's low throughput. Need a different setups. So in that sense, it's low throughput. And obviously it's poor spatial resolution. Because of this, as I explain later, the signal interpretation is a bit tricky. In terms of temporal resolution, it's rely on the sensors. So because the photodetector can perform very quickly, so depending what type of sensor, biosensor you will use, so temporal resolution will be determined. So in terms of signal interpretation, so because of this sort of a weakness, you need to have some controls, which I will explain next slide. So you might have heard of isostatic signal. So it's a bit technical word, but when you want to do fiber photometry, so this is essential. So this is sort of a negative control. So why we need it? So let's imagine this situation. So you have optic fiber and then there are sample, but we do this in the live tissue, so the actual brain, and the brain tissue is very soft. So if animal move, So obviously, so distance can change like this. And then, so obviously, even G-camp signal didn't change, you can see some artificial fluctuation of the fluorescence. So you need to correct this sort of motion artifacts. So therefore, people are applying the monitoring isospecific signal. So how it works. So this is the absorption and the emission spectra of the G-camp. So when you monitor G-camp signals, typically we apply around 470 nanometer light to activate, excite. And then we typically monitor emission signals at the 525 nanometer. So this is a typical G-camp monitoring. So isospecific signal can be monitored by applying another light, which typically are 405, which is very tail of the G-camp. So basically, you can't excite the G-camp. So thereby, so when you monitor G-camp, so when you only use excite, so 470 nanometers excitation light, you can get the two signals. So one is a calcium dependent, so the G-camp signals, but also you get the motion artifacts, which is a calcium independent signals, which you wouldn't have. So therefore, you need to have this isospecific signals, which is 405 nanometer, which provides calcium independent signals. So after collecting data, so you need to reject, eliminate these calcium independent signals, which is very important in these fiber photometry experiments. And some people using different flow holes like TD tomato, or some people do separate experiments by expressing GFP to get the so-called stable, not the dynamic signals. And unfortunately, if you read some papers you never see such a control signals. So in that case, you need to be very careful to interpret their results, even in the published papers. Okay, so that's the sort of outline of fiber photometry. Okay, so next I will move on to the little bit more technical aspects. So if you are interested in the fiber photometry, and if you want to set up the photometry, I will give us some overview of how to do it. So before that, there are two review articles. So one article published Neuron 2023. Almost at the same time, we also published some perspective technical review articles. So I strongly recommend you to read these two review articles to better understand the fiber photometry. Okay, so as I mentioned, so you need two major components, so biosensors and optical setup. So first I will talk about biosensors. So there are a bunch of wide range of genetically encoded sensors. So if you read these review articles, so you can see comprehensive list of sensors. So I just pick some of them here. So if you want to monitor calcium, so obviously G-camp is the most popular genetically encoded sensor. So the latest generation is called JG-camp8, and there are three variants. S is slow, but which provide a better, stronger signal, and medium and fast. So the kinetics is different. And if you want to combine another experiment using different wavelengths, you might want to have a red-shifted calcium indicators. In this case, people often use R-GECO-1A, or this is not published yet, but some group of scientists developed a pinky camp, which seems to be very promising. So for example, if you want to combine these two genetically encoded sensors, you can monitor signals from two distinct populations by expressing these sensors. Or you can combine with optogenetics. And you might also interested in the neurotransmitters or neuromodulators or neuropeptides. Actually, there are a wide range of sensors. So basically, there are three major categories. So SEM4 series, which developed by Jenny project. So recently they upgraded their sensors. So this is a glutamate sensor, this is a GABA sensor. So this is a version 4 and version 2. So you can see bioarchives, and probably you can also access some plasmid or even viral vectors. And Yuron Lee group developing GRAP sensors. So in this particular science paper, they developed the GRAP sensors for several neuromodulators, but also they have a GRAP sensor for dopamine or acetylcholine, many other neuromodulators. And Light series, which originally developed by Lin Tian and also Tommaso Patoriaci. And Tommaso Patoriaci get a position in Zurich, and now his group developing a wide range of light series, light protein sensors. So, for example, recently they developed the sensor for noradrenaline, and also they developed acetylcholine sensors. So these board signals are what we are using in our project. So there are sensors for acetylcholine as well. So basically, there are a wide range of sensors. And so how you can introduce these sensors in the mouse brain? And so if you particularly interested in certain cell types, you can think of two approaches. So one approach is using transgenic approaches. So you can cross two mice, and then you can express some sensor like a GCAP. So in this case, you need a CRI-dependent driver mouse, CRI driver mouse, and also you need a reporter mouse, which can express reporter. Or, so this is more popular, so you have a CRI driver mouse, which can target certain cell type, and then you can inject the CRI-dependent adeno-associated virus into certain brain region. So there are pros and cons. So if you take transgenic approaches, so obviously it becomes reliable and reproducible, and you can express some reporters, across that particular neurons, across the brain or even the body, and you don't need to do any surgery. On the other hand, if you are really interested in certain brain regions and you wouldn't express GCAP in different brain regions, so this approach may not be suitable. And also, you need to maintain two mouse lines, so it becomes more expensive. And the major constraint is that there are fewer reporter mice. So you have less options. And also, you can't control when you want to introduce gene expression. So depending on CRI, so gene activity of this CRI driver mouse, so you will get expression even from early development, depending on genes. And if you take viral approaches, so of course there are several strengths. So it can be selective and also cheaper compared to transgenic approach. And also there are a wide range of viral vectors. Many of them are commercially available. On the other hand, because you need to do surgery, so the expression pattern can be variable. And also, because you need to inject the virus into the mouse brain, so the neurons or cells get infected, so which could induce some sort of inflammatory responses. So that's a sort of a potential issues. So of course, there are no perfect solutions. So depending on what you want to do, and what sort of options available, you can make a decision to target a certain cell type. Okay, so far I talk about biosensors. So okay, let's look at the more hardware aspect. So when you want to introduce, so you need to make a decision. So I hope you get this analogy. So you need to pick either blue pill or red pill. So if you take a blue pill, so basically you're happy. So you don't need to open the black box. Okay. But if you take a red pill, so basically you will open up the black box and you have a little bit tougher life because you need to realize the reality. So I will explain that. So in terms of blue pill options, there are multiple companies selling the sort of system for photometry, like Tucker Davis, TDT, or Dorek. So these are probably the popular companies, and also Plexon or MVF Bioscience also selling photometry system. So so you have some budget, and if you want to save your time, so these are good options. So we never use this system because we took the red pill. So we decided to build our own system by purchasing multiple components from multiple companies. So Thorabs, Newport. So the photodetector was purchased from Newport. And to control these devices, so we purchased some device from National Instrument. So in this particular study, because we wanted to combine electrophysiology, so we combine with the INTERM system, which is suitable for electrophysiology. So if you are interested in fiber photometry alone, so you don't need this INTERM system. And so these are sort of key components. So obviously you need optic fiber, which will be implanted to the brain. So here, and you need a patch cable here, which deliver light, and also you can collect the light. And you need the light sources. So you need LEDs. It can be laser as well. And so depending on the configuration, you need appropriate mirror, dichroic mirror or filters. And then, so the signal will be captured by the photodetector. Okay, so basically light comes through here, then excite the fluorophore, then you can collect the light, then you can detect the light. And then, so these LEDs and the photodetectors are connected with the data acquisition system, like this. Then you can control LED and then you can monitor the signals. So this is a much more realistic view. So this is a patch cable, and then there are two LEDs. So these LEDs, and there are multiple filters and mirrors here, and then photodetector looks like this. So this looks complicated, but the good news is that Thorabs actually selling much simpler components. It's much more like a cube-like structure, modular system. So you can put a mirror or filter, and then you can connect LEDs, and then you can connect a photodetector, and then you can connect a patch cable here, and then so this implant will be go to the brain, the mouse brain. So actually, our second generation of system utilizing this system. But of course, you still need to develop, build your own system. Okay, so this is much easier to build. Okay, so now let's assume that you set up the system. So this is how you can do experiment. So first of all, you need cream eyes, and then you need to inject a virus, let's say, so to express GCamp. So this is a week 1, and then so we typically incubate around 3 weeks, and then week 3 we implant the optic fiber. So in terms of optic fiber, there are two major options, I think. So 200 or 400 micrometer diameter. So given the GCamp 8, I believe that 200 may be sufficient and also less invasive. So, and if you have a problem with this thinner optic fiber, you may consider 400 because thicker optic fiber can collect more light. So you might have a better chance to get the signals. And then another recovery period. After another recovery period, you can do habituation, and then you can start your own experiment. So this is sort of a typical experimental workflow. And I mentioned that the data interpretation is not straightforward. So this is how it looks. So I took this image from this technical review article published in Neuron. So there are multiple steps. So this is our typical pipeline. So first of all, you need to reconstruct signals because we need to use the two light sources, 405 and 470. So the light will be alternated. So therefore, you need to separate these two light signals. So what I mean by signal reconstruction. Then you can apply filter to get rid of high frequency noise to make signal cleaner. And because this is fluorescent imaging, sort of, so you will get photo bleaching. So as a function of time, you have a gradual decrease of signals. So to correct the baseline signals, so you can basically consider this photo bleaching effect. So typically we consider exponential curve to estimate this photo bleaching effect. So thereby signal become flatter. And then, so as I mentioned, so using isoscelistic signals, you can correct the motion artifacts. So typically we linearly scale the control signals and we can subtract. So it's a bit complicated. And then finally, you can normalize signal, which allow us to compare the signals across experiments, between the recordings or across animals. So these are typical workflow. So whatever you establish this data analysis pipeline, what I want to recommend you is to check the signals, go back to the sort of intermediate signals to see what sort of signals you are dealing with, or whether you have a photo bleaching or not. In some cases, you might not see massive photo bleaching. In that case, you can skip this step. And also, this is another important aspect. So the isoscelistic signal should be correlated with the real, let's say 470 nanometer signals. If both signals are capturing the non-specific motion signals. So you might be able to see this type of linear correlation between two signals. If so, so this motion correction process will work. But if you apply very weak isoscelistic signal, if you get the very weak isoscelistic signals, you might not see this type of linear correlation. So in that case, you might have a problem with the motion correction. So again, so the checking these sort of intermediate signals are very important. And also when you establish data analysis pipeline, you will be able to explain why you doing these steps. So by checking these signals, you have a better understanding of what you need to do and also what you are doing. So this has been a hurdle because it typically requires some coding. So we use in MATLAB but of course Python can also deal with. But the good news is that these days, using AI, like ChatGPT, Claude, or Gemini, actually they provide really nice scripts, which you can use almost directly or just minor changes. So the combining some programming language with AI, so you can deal with the data processing. And also there are publicly available softwares, sort of functions, codes as well. Okay, so that's a sort of general workflow. And so here I just briefly explain sort of a little bit more advanced technology, so-called tapered optic fiber, which we also use them. So originally, so this technology was introduced 2019. So the fiber actually tapered, it's more sharpened. And by changing the angle of laser penetration, so you can shine laser only the sort of base level here, upper part, or if you penetrate the laser go straight, you can illuminate only the tip. So more like this. So basically you can scan the tissue along this taper. So thereby you can get the depth resolved photometry signals. Guess I will get back to this technology later. So the point is there are some advanced approaches in photometry. Okay, so let's summarize so far. So first I introduced the principles, so how photometry works, and what sort of components you are required. And also I briefly mentioned the history of this technique. And also I compare with other methods. And it's very important to have some control when you do the photometry experiment. And also I briefly summarize what type of sensors are available. And you can decide whether you can take a blue or red pill options. So if you take a blue pill, it's easy to implement, but you need to pay more. If you take a red pill, it's affordable, but you need to build your own system. And I also outline the typical experimental workflow and also data analysis pipeline. Okay. So still have 10 more minutes, I think. Okay. So now I will talk about how my group has been applying the fiber photometry. So in our lab, so we are primarily interested in two major aspects. So the one is sleep, the other is Alzheimer's. And so I listed some applications. So for sleep research, we monitor the neural activities, of course, but also, so the amazing thing about photometry is that you can also deal with the astrocyte, also non-neuronal cells. And in the Alzheimer's research, so we monitoring cholinergic signals, and now we started to look at the microglial calcium signals as well. And at the end, I also mentioned about the plaque, amyloid plaque monitoring using photometry. Okay, so this is sort of general introduction of our research. So we are interested in brain states as brain states. So if you think about the conscious state or unconscious sleep state, our brain waves keep changing. So during wakefulness, you will see typically a very flat and a very fast brain waves. So these are EEG, electrophysiological signals. And when you fall asleep, brain wave become larger and slower. So we have a different brain states. And these brain states are associated with different behavioral or arousal states. So that's what we are interested in. And so these brain states can change in minutes or hours, but so this structure of brain state can also change over the age. So this can be the months or years. So our questions, major questions are, so first of all, we want to better understand how these brain states are organized and how these brain states affect the brain functions. And so we are particularly interested in this at the moment, the mechanism, how different brain states or sleep states are regulated. And the fourth question is, this is related to disease. So the targeting certain brain states. So we ideally want to modify disease pathology. So these are sort of major sort of goals which we are aiming. So in terms of sleep, so there are big technological challenges if you really want to understand sleep. So if you think about sleep-wake cycle, so this transition can happen in minutes or hours. But if you think about brain waves, like oscillations, you can see, this oscillation can happen within millisecond order or within second. So you need to deal with multiple time scales from millisecond to hours or even days. And if you think about spatial scales, so these are sort of a summary of which brain regions are regulating non-REM, rapid eye movement sleep or non-REM sleep. So as you can see here, so it's widely distributed. So you need to deal with entire brain, and also not just neurons. Non-neuronal cells also contribute to the sleep regulation. So you need to deal with both major cell classes. And some chemicals like acetylcholine and norepinephrine also very important to understand the sleep regulation. So you need to deal with some chemicals. So spatial scales also massive. So it's very challenging. So what we are doing is, of course, we can deal with everything. So this is our approach. So we combine electrophysiology or optochemogenetics, and then, of course, the fiber photometry. So in next several slides, I will briefly introduce how we use the fiber photometry to study the sleep. So in this particular study, published 5 years ago, so we are interested in brainstem. So we wanted to understand how population brainstem regulate the sleep. And so here in this particular experiment, we targeted cholinergic neurons in brainstem. So by expressing GCAP, we monitor population activities. So as you can see here, so this summary. So during REM and wakefulness, you will get the stronger signals here. So this red and green. But during non-REM sleep, cholinergic signal is weaker, smaller. So this is sort of a state-dependent signals. This is one way to apply photometry. And in these two papers, we combine the electrophysiology with fiber photometry. So because so we are particularly interested in certain brain waves, so-called Pontin waves or PGO waves, which is often these waves can be seen typically during REM sleep. So it's like a biomarker of REM sleep. And because we wanted to better understand these waveforms, brain waves, we needed to combine both technologies. But there are some advantage and disadvantages. So if you think about electrophysiology, you get a very fast signals, but you won't tell which neurons are active. But if you use a fiber photometry alone, you can get the cell type specific activities. But if you use the GCAP is very slow. So therefore, temporal resolution is very poor. So therefore, we decided to combine these to complement these both advantages and disadvantages. So approach was very simple. So we basically made a sort of a hybrid probe, this electrode, and then we just simply glue with the optic fiber. Then, by expressing G-camp in cholinergic neurons, we can monitor photometry signals. So these are cholinergic neuronal activities, but also we can monitor Pontin waves. So these red spikes, these are Pontin waves. So actually, when Pontin waves happen, we typically see big cholinergic activities. So this is the summary. So time 0 is an onset of P waves, Pontin waves. So in both states, non-REM and REM, we got the big calcium activities. So interestingly, during non-REM sleep, calcium signal gradually decrease and then P wave appear, but also cholinergic signal also slightly increase a little bit. And then entire cholinergic signal gradually increase. So there are clear sort of state-dependent cholinergic activities across sleep states. And so not just neurons, so we also monitor astrocyte calcium signals across sleep-wake cycles. So the dogma was that the intracellular calcium signal in astrocyte tells us sleep need. So as we keep waking, intracellular calcium signal in the astrocyte gradually increase, and then if animal fall asleep, calcium level decrease. So we tested this idea by using fiber photometry. So what we did was expressed another calcium indicator across the brain, and then we penetrated the optic fiber multiple brain regions. So somatosensory cortex, hippocampus, lateral hypothalamus, and some Pontin lesions. So if you look at the cortex or hippocampus, indeed calcium level decrease as this theory predicted, like this. However, when we look at the calcium signals in astrocyte in the hypothalamus and the pons, actually we didn't see such a reduction. So rather, in pons especially, we see slight increase in calcium in astrocyte. So having said that, so this theory can be applied only cortex and hippocampus, but not for the hypothalamus and pons. So basically, this theory need to be sort of updated. So this is another application of fiber photometry. So that was about sleep. We also studying the Alzheimer's. So what we interested in this. So by controlling brain activities, ideally we want to stop or intervene the pathology. So we have 3 major activities. So we want to identify some targets or biomarkers to manipulate. And so we want to combine some optogenetics or whatever to modify disease states. And another pillar is to develop some technology to monitor amyloid plaques or pathological the state in the brain. So I briefly mentioned about these 2 aspects. So these 2 projects, just 2 slides. So here we monitor acetylcholine in hippocampus. So using Alzheimer's mouse model. So here we implanted the hybrid probe. So it's optic fiber and also electrode. And here we express the cholinergic sensor. So this is just an example. So these are cholinergic photometry signals. At the same time, we monitor electrophysiological signal in hippocampus. So this is a spectrogram across sleep-wake cycles. So this is just a demonstration what we did experimentally. And another, so this is a final slide. Recently we managed to monitor the amyloid plaque signals in the freely behaving Alzheimer's mice. So here we combine this advanced technology, so-called tapered fiber, by collaborating with the Italian group. So setup is like this, a little bit complicated, but basically we sort of scan the laser along the taper to monitor plaque signals. And to label the plaques, we use this chemical, so-called methoxyX04, which is the BBB crossing chemicals, which can bind to the plaques in the brain. And then, so this is a fluorescence molecule. So by shining, in this case, 405 nanometer light, you can see the plaque signals. So here, so this is a scanning depth. So this is a deeper region, so this is the upper region, like this. And when we injected this methoxy, so we saw gradual increase in this fluorescent signals, but also we see some sort of heterogeneous signals in the Alzheimer's mice. So this is sort of a quantification. But if we did the same experiment in the wild type, obviously we didn't see anything. So this approach allows us to monitor amyloid plaque signals across the depths, even in the freely behaving animals. Actually, this mouse actually moving. Okay, so that's applications. So let's summarize. So first part, I introduced photometry. Then in second part, I give some technical guide to set up the fiber photometry. And then finally, I introduce some applications. I hope that I convince you that the photometry is very powerful to deal with multiple signals in the brain, especially in vivo. So you can monitor not just neural activities, but also you can monitor non-neuronal calcium signals. And now you can monitor neuromodulator signals, but also you can monitor some pathological molecular signals as well. Yes, with that, I'd like to thank to my current and former lab members and also collaborators, and also funding agencies. So with that, yeah, thank you for your attention.